Hi everyone. Today we're going to talk about single nucleotide polymorphism, or SNPs for short. SNPs are the variation in a person's DNA base like A, T, C, or G at a specific location in the DNA sequence. These variations happen naturally and can help forensic scientists compare an individual's DNA to a reference sample. SNPs are found about every 100 to 300 bases and can tell us a lot about a person, like their traits, ancestry, and identity. They're useful in forensics when other types of DNA evidence aren't strong enough to convict, or the DNA is destroyed before being able to convict.
This is the history and development of SNPs. SNPs was a forensics tool that was developed by multiple scientists together and institutions. One of the first, quote-unquote, main contributors was Leroy Hood in 1980. Him and his team helped create automated DNA sequencing, which is the foundation for SNP analysis. Later in the years, the Human Genome Project, which ran from 1990 to 2003, made SNPs more valuable by mapping the entire human genome. SNPs started being used for forensics analysis in the late 1990s to early 2000s, but they weren't accepted in court until 2019. That year, a Dutch court used SNP evidence in a sexual assault case. The original DNA was inconclusive, but SNP profiling helped identify the suspect and led to the person's arrest. This case marks the first time SNPs were accepted as legal evidence.
The SNP process involves four main steps. Step 1, sample preparation and DNA extraction. A sample of the blood, hair, tissue, or etc. is collected. The DNA is extracted and checked to make sure there are enough SNPs to ensure its high quality. Step 2, PCR amplification. PCR, or polymerase chain reaction, is used to make copies of the DNA. Primers target the sections that contain SNPs so they can be looked at. Step 3, genotyping. This is where the actual SNPs are identified. Different methods can be used, including real-time PCR with fluorescent dyes, snapshot reaction, which uses single base extension, or SNP microarrays, which use chips to scan for many SNPs. Step 4, data analysis. The results are analyzed to figure out what SNPs are present and what they say about the person. This can include identifying physical traits, ancestry, or even matching them to crime scene evidence.
So when and why do we use SNPs today in forensics? SNPs are useful when working with low quality or old DNA, like in cold cases. They also give investigators more information, like a person's eye color, hair color, skin tone, and ancestry. SNPs help when standard STR testing doesn't work or gives unclear results. This makes them super valuable in forensics investigations, especially when we don't have a full DNA profile.
To make strong individual match using SNPs, scientists need at least 700 SNPs and a segment that's 7 centimorgans long. This ensures the DNA match is reliable enough to use in court or for conviction. To wrap this up, SNPs are a powerful tool in forensic crime. They help identify people, solve crimes, and even uncover traits from just tiny pieces of DNA. Thanks to advances in science and tech, SNPs are now accepted in court and can be used alongside other forensic methods. Thanks for watching our presentation on SNPs. We hope you learned something new about how this small genetic difference can make a big impact in forensics.